can access it. Please, over to you, Bruce.
Thank you very much, Vitaliy. Thank you for the invitation. I'm going to share my screen, and then you can verify that you can see it. Okay, can you see that screen all right? I'll move this bar.
We can see Bruce, yeah.
Okay, perfect. So thank you, everyone. First, my disclosures. I have a financial interest due to intellectual property and patents in the field of cell and gene therapy, and that conflict is managed in accordance with University of Pennsylvania policy and oversight. So I thought I would divide today into 4 sections. We'll talk about the beginning, how to make faster and stronger T cells, attacking more challenging cancers, and finally, the challenge of scaling to treat more patients. So if we go back to the beginning of where chimeric antigen receptor T cells came from, it was in the 1980s when the structure of the T cell receptor and the B cell receptor was being elucidated, and there were 2 separate groups that published these papers. And the constructs used on the one on the left are shown, and some of the functional data on the right. But what really struck me in reading and rereading these pictures is in the discussion, they say in the future it might be possible for T cells recognizing any antigens without MHC restriction to be produced by the technique described here, or this approach can be exploited to direct cytotoxic T cells to kill tumor or virally infected cells. So even back then, they were thinking about therapeutic applications. Now, what exactly is a CAR T cell? Well, let's go back to what the chimera is. It's a mythical beast with the body of a lion, the head of a goat, and the tail of a serpent. In our case, we're constructing a molecular chimera where the outside of the cell, the extracellular, is an antibody or other ligand binding domain, and intracellularly, there are signaling domains that deliver signals to T cells. So this is a molecular chimera. Now, this is the first known depiction of the Trojan horse. We need a Trojan horse to carry the genetic material for the CAR inside the cell. We're using a lentiviral vector, essentially disabled HIV. So how this comes together is we have a T cell with its native T cell receptor. It receives via lentiviral vector RNA reverse transcribed into DNA permanently integrated into the genome that is expressed on the surface of the T cell that still retains its native T cell receptor, but now the CAR redirects to find tumor and kill tumor cells. So that is a cartoon version. This is what things look like under the microscope. We've colored tumor cells green with GFP. The CAR T cell is on the bottom left. And what you'll see over the course of 8 seconds is the engagement, the recognition, activation, and secretion of perforin and granzyme, puncturing holes in the tumor cell, and that tumor cell explodes. Now, this is a living drug. So that CAR T cell will go on to that next tumor cell and kill that next tumor cell. Just taking a look at where we come from with a endogenous T cell receptor that must recognize antigen in the context of major histocompatibility complex. So it's tissue type specific. Now you can isolate tumor specific T cell receptors and insert them, but they're still restricted by MHC. These T cell receptors do recognize intracellularly processed antigens. A CAR recognizes surface antigens and again is MHC independent. Now, I showed you the innovation trigger in the 1980s, and there was great excitement and start of clinical trials in the late 1990s and in cancer, but those first trials in cancer were disappointing due to poor T cell engraftment. And so we entered a trough of disillusionment. I think we're now somewhere between the slope of enlightenment and the plateau of productivity. How do we manufacture these CAR T cells? Well, we do it outside of the body. We collect white cells via leukapheresis. We can enrich or purify the cells that we want that are the T cells. We deliver that genetic material using the lentiviral vector. We stimulate T cells with an artificial dendritic cell. It is a magnetic bead to which we conjugate two antibodies, one directed against CD3 and one directed against CD28, thus delivering efficient co-stimulation. We'll grow the cells up in the bioreactor traditionally for 9 days, but I'm going to show you a different process. At the end of that 9 days, we remove samples for quality control testing to address safety, purity, potency, and identity. Once that testing is performed and quality assurance reviews, we can release the product that had been cryopreserved at formulation. So because T cells can be cryopreserved upon collection and the final product, the manufacturing can occur anywhere in the world. In 2010, we started our first clinical trial in cancer following trials that we did in HIV in the late 90s. And this first trial in cancer was targeting CD19, present on T-cell leukemias and lymphomas. The first 3 patients had chronic lymphoid leukemia that was refractory to all available treatments, and they were not eligible for a stem cell transplant. This is our first patient. Up top, you can see everything brown here is leukemia. 6 months later, it's clear. And in the first 3 patients, between 1.3 and 3.5 kilograms of leukemia was obliterated in the several weeks after transplant. Now, after treating patients with chronic lymphoid leukemia, we wanted to move to acute lymphoid leukemia. But this is a much more rapidly growing cancer. These patients are essentially falling off the end of the world and progressing very quickly. We opened this trial in children at Children's Hospital of Philadelphia. This is the first patient who was 7 years old in this picture, was diagnosed when she was 5, relapsed twice, came to Children's Hospital, received high-dose Cytoxan and clofarabine, a regimen that will kill an adult. Only about a third of kids make it through. She did, but she still had lesions in her kidney, liver, and spleen. Was not far from her demise. She received her CAR T cells, and 6 days later, we're looking here. This is leukemia, and with an anti-idiotypic antibody, we're looking for the CAR here. So you see all this leukemia in the bone marrow, and then at day 23, you see this massive expansion of the CAR T cells, and by measurable residual disease, the leukemia is gone. Well, that's the good news. The bad news was severe cytokine release syndrome that we saw in this patient that was refractory to steroids, to Enbrel, and you see her IL-6 levels were high, 10 times higher than we had seen in any adult. And she was given tocilizumab, a drug that at the time was recently approved for arthritis, here used in a leukemia patient to ameliorate severe cytokine release syndrome. So we learned a lot from her. And she was the first child in the world treated, but certainly not the last. Based on pivotal clinical trial data, we had approval of Kymriah in the US August 30th, 2017. We now have in total 7 CAR T products approved in the US, most of those approved in Europe and other jurisdictions. We have tumor-infiltrating lymphocytes approved in the Netherlands, US. We have a T-cell receptor product, an allogeneic T-cell product. We have 4 products approved in China. We also have gene-modified stem cell products approved for rare diseases and for sickle cell and thalassemia. So this is really a burgeoning of this era of synthetic biology. Now, one of the first three adults is shown on the left. He is now actually more than 14 years cancer-free. Emily, the first child, is 12 and a half years cancer-free and a sophomore at the University of Pennsylvania. But not all patients respond. So how do we see more of this? How do we increase response rates, approvals, and patient access? In other words, how can we teach every T cell to be all that it can be? Well, let me share with you some designs and faster and more powerful cars. This is from a review that we wrote a few years ago on different models of cars. And I won't go through all of them. I'll just reference the article for you, but I do want to talk about the variety here. And I started collecting reports of how you make more potent CAR T cells, and pretty soon I had a long list of 50 and growing. Now, if you're wondering which combination would be best, or whether it's 2 or 3 of these modifications, whether it's a gene inhibition, a cytokine, or gene overexpression, that's a very large number. So we need better ways to screen these in animal models and potentially in humans. Okay, now to faster and stronger cars. And this is shortening the ex vivo manufacturing to 3 days. And one of those car models I showed you was a truck, which was T cells unrestricted by cytokine killing. In our case, we're using IL-18 to enhance the potency of the CAR T cells. So how does this truck work? Well, you take the CAR, and we're using a CAR with a co-stimulatory domain of 4-1BB and a stimulatory domain of CD3 zeta, and you include in the construct an IL-18 that is secreted locally when the CAR T cell engages. Now, this approach was validated by preclinical data from our group and from other groups, and the clinical trial opened 2 years ago. Now, I showed you there are 7 approved products. 5 of those are targeting CD19, many other clinical trials targeting CD19. So the important thing I want to point out to you here in this clinical trial, all of our clinical trials address relapse and refractory patients, but in this one, because of the availability of CD19 CAR T cells, we included an eligibility criteria that patients must be relapsed after or refractory to a prior commercial CAR T cell therapy if they had been indicated for that by the FDA label. Now, we started at a very low dose, not per kilogram, but 3 million total CAR T cells. This first patient had received 9 prior lines of therapy, including radiation, an autologous transplant, a commercial CAR, a bispecific antibody, and look what happens with 3 million total T cells, a dose that we would give a couple of mice. This is before and after, before and after, before and after. And she's been in a continuing metabolic response. And this was a dose that we thought too low to see anything, but had been requested by the FDA for safety reasons. Now, in the larger cohort of 21 patients, you can see the overall response rate here of 81%, a complete response rate of 52%. The duration of response is shown here on the right. This is looking at responses by non-Hodgkin's lymphoma subtype. There's not really a difference, although there are small numbers. The tumor volume change is shown here on the right. If we look at the peak in vivo expansion by dose level, there doesn't seem to be a change. And we did see a couple dose-limiting toxicities at dose level 5, so we have dose de-escalated. Now, this is important. Remember I said this is a living drug. there's no apparent difference in response rate by the dose level. So what counts more is quality, at least over this range of dose levels. But interestingly, what does affect response and peak expansion is the co-stimulatory domain that the patients have received in their prior CAR, with a higher response rate in patients who had previously received a CD28 CAR like Tecartus and Yescarta compared to a 4-1BB CAR. The safety shown in the middle, the responses by CAR subtype are shown on the right-hand side. So to conclude the study that's still ongoing, this is the first study using an armored CAR-T product secreting IL-18. Treatment is feasible, well-tolerated. We have durable responses. The peak expansion could be affected by the prior CAR type, and IL-18 may enhance CAR-T cell efficacy by intrinsic or extrinsic mechanisms or combination. So now I'd like to move to attacking more challenging cancers and just using as an example glioblastoma. Now, glioblastoma, aggressive standard of care includes resection, radiation, and chemotherapy, but there is essentially a 100% recurrence rate. And when recurrence happens, no treatment improves overall survival. So there's a dismal prognosis with median overall survival on the order of a year, and median survival from recurrence is just 6 months. It's also highly infiltrative, and that really makes complete resection impossible. And when thinking about immunotherapeutic approaches, it is molecularly heterogeneous. Now, there were 3 papers that occurred the same week in March 2024 that drove home the importance, we believe, in solid tumors of location, 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 as they say in real estate. And so these 3 papers were from City of Hope, from Massachusetts General Hospital in Boston, and from Penn. And in our trial, we infused the CAR-T cells through an Ommaya reservoir, which is standard of care for delivery of chemotherapy in these patients. These papers, besides location, all had one thing in common, which is a combination approach. In our case, targeting 2 separate antibodies, in Mass General, targeting 2 antibodies, one by a CAR and one by a bispecific antibody. So that's another important point, that going back to our CD19 CARs, CD19 is pretty much a perfect tumor antigen because it's expressed on tumor and healthy B cells, but it's not expressed on other T cells. And virtually all solid tumors, or all solid tumors, I think we can say, they are molecularly heterogeneous. And the other challenge is that the antigen may be expressed brightly on tumor, but also dimly on other tissues. So one has to worry about toxicity. And this is just a snapshot from one of our patients. This is pre-infusion and post-infusion, where you see within 48 hours necrosis of this central lesion. Now, we have conducted over the past decade 2 other clinical trials in glioblastoma administering IV, and the engraftment is seen here. Now, when we administer through the Ommaya reservoir, you can see 100-fold higher engraftment where you want the CAR-T cells to be. So in this trial, we have a bicistronic CAR targeting IL-13 receptor alpha-2 and EGFR, epidermal growth factor receptor, and amplified recurrent GBM. There is significant neurotoxicity in these patients. That's not unexpected. We have to think about the manufacturing turnaround time and the rapidity of the progression in these patients. And We have clinical and imaging responses that are pending and tissue analysis and other correlative studies that are ongoing. So now I'd like to move to the challenge of scaling to treat more patients. And this is a challenge of scale. So we have approvals in leukemia, lymphoma, and myeloma for CAR T cells. What happens when we have approvals in solid tumors? That's 10 times as many patients. And when we have approvals in autoimmune disease, again, 10 times as many as in heme malignancies. And then thinking more broadly in fibrosis diseases, the engineered stem cell approaches in hemoglobinopathies and rare diseases, there aren't enough humans on Earth to do the ex vivo manufacturing of all of these therapies, let alone the infrastructure. Now, there are allogeneic approaches being developed and also induced pluripotent stem cells. But what if we could leapfrog those approaches with an off-the-shelf autologous approach? So what does that mean? Well, if we combine the technology of CAR T cells with the technology of the COVID vaccines, that's what I'm showing you here. Now, the lipid nanoparticle is encapsulating messenger RNA, but instead of COVID SARS spike protein, the mRNA is encoding a fibroblast activation protein targeted CAR. This paper in induced model of cardiac fibrosis shows the potential of this approach. Now, naked lipid nanoparticles will traffic to the liver. We do not want to have the targeting to the liver. We want to target T cells. So to do that, we're conjugating here to the surface of the lipid nanoparticle an antibody that directs the lipid nanoparticles to T cells either through CD5 or CD8. So this is the lipid nanoparticle. It's a proprietary lipid formulation, a targeting antibody, and RNA payloads. And you can think of different approaches in oncology, in autoimmunity, in fibrosis, or blood-brain disorders. I want to share with you some of the optimization that was done on the lipid formulation here. The COVID LNP, otherwise known as the tool here, is shown here. So look at the ratio of the liver to the spleen. Remember, we want to go to the spleen. Here is an antibody-targeted lipid nanoparticle. And that ratio is a little bit different, but not much. And here is a capstan formulation lipid. And you can see detargeting of the liver and targeting the spleen. To look at that in a graphic form, on the left here, this axis is liver CD45 cells. So lower left is better. This is an M-cherry reporter gene. This is that CD5 targeted capstan, and this is the COVID vaccine. And then on the spleen here, upper right is better. So here is the COVID vaccine LNP, and here is the targeted capstan LNP. And in humanized mice, targeting CD19 with a CD8 targeted lipid nanoparticle, we see rapid B-cell depletion within hours. So this is 3 hours after intravenous administration of targeted lipid nanoparticles with a CD19 CAR. And so that B-cell depletion correlates with the CAR expression in CD8 cells. And in non-human primates, we see this is multiple administrations. This is looking at the CAR-positive cells at different doses. And you can see this dose response and increasing area under the curve with each subsequent administration. This is in CD4 cells. Now, here we're targeting CD8, so we don't want it in the CD4 cells. Some activated CD8s do express CD4, so there is some minimal expression here. And this is data that is really, really exciting for us. And this is showing deep depletion of B cells, not only in blood rapidly, but also in tissues. So up top is the different dose level and depletion of B cells in blood in these monkeys. And this is the spleen. And we also see that dose response of the tissue depletion in spleen. And this is important not only for oncology, but also autoimmunity to achieve that reset that has been seen by groups using CAR-19 targeted T cells. And that deep B cell depletion is achievable with 2 doses up top in the blood and here in the spleen and the bone marrow and the lymph node. Now, after administration of those targeted LMPs, the B cells that come back are of the naive phenotype, and that's exactly what we want. So these mature cells, now this is not an autoimmune model in NHP, but we're using the phenotype of the B cells as a surrogate for immune reset. So to summarize this in vivo cell engineering with targeted mRNA LNPs, we have the ability to reprogram specific cells to have rapid T cell engineering and B cell killing within hours, not only in the blood, but also in secondary lymphoid organs. And this is potential application in, this should say, measurable residual disease and designed to facilitate access to CAR T cells for a large number of patients. I'll just also say really quickly that we're looking at what does predict response, and we're looking at everything from the characteristics of the patients, of the apheresis, elements during the manufacturing, the infused product and long-term product to use artificial intelligence and machine learning to tease out those factors like early cytokine response that may predict a response. So just summarizing this predictive biomarker data, it allows us to look at the underlying T cell biology that is associated with anti-tumor responses. And we know that ex vivo expansion during manufacturing is associated positively with a good clinical outcome, as well as post-infusion expansion, and I'm showing you the cytokine profiles here. Now, just coming back to the access, I just want to mention some of the barriers that patients face, financial, logistics, caregiver, having an advocate, and healthcare providers. It's referrals, institutional infrastructure, logistics, and clinical capacity and expertise. And for the manufacturers, it's the technical infrastructure, talent, and onboarding clinical sites. So some critical path issues for wider patient access are enhancing potency, especially for solid tumors, where we've not seen the dramatic and striking results that we have seen in the hematologic malignancies. I showed you an example of using armor with IL-18 to enhance potency. There's other work with switches and combination therapies. Ex vivo manufacturing is complex. These are cell-based living drugs using viral vectors. How can we implement automation, shorten the ex vivo culture and the vein-to-vein time, or implement in vivo? We need, in this new pillar of medicine, education and training at all levels. So Vitaliy mentioned the past president of the International Society for Cell and Gene Therapy. This is an educational forum, so appreciate the chance to present to you. We have to work on access disparities and geography, race, income, and caregiver availability. We have to recognize that these therapies are currently financially complex. Affordability is a challenge. can we implement value-based payment that links price to outcome? And I shared with you very briefly how we might think of bringing in predictive biomarkers. So it truly does take a village to make these trials happen. This is the flash mob at the Cancer Center on the day of FDA approval. Sadly, I'm not in this picture because I was in Boston giving a talk on CAR T cells. I'd like to say that I'm a representative of the large teams of teams at the Center for Cellular Immunotherapies at the University of Pennsylvania, collaborators at Children's Hospital, philanthropic, government, and industry funders, collaborators at the FDA, especially thank our study participants. This is the CAR-19 IL-18 acknowledgement slide, the in vivo T cell engineering slide. And Vitaliy mentioned it up front. Now, this is an experiment, and I'm going to experiment on you to see if this works. So there's a story behind this that I can go into later or another time, but I met a very talented singer-songwriter, dancer, musician who hails from Cork, Tripsy. Some of you, or most of you, hopefully know where that is. And we wrote a song together about CAR T cells. So with your indulgence, I'd like to play that for you now. So this is Mags McCarthy. It's actually her birthday today, and she has a new song, not this song, but another song that has just come out on Spotify and on the radio. So here we go.
I walked that road, saw that look on your face. You're gonna say you came back. We all fear that place. But don't you worry, you soon see the light, and we'll never. We keep on fighting, that's right. There's some people, there are options to choose. Did you hear about this new world to you?
Holly tells me the song is, the audio is gone. Is that true?
Hi, Bruce. Unfortunately, we cannot hear the audio. But thank you for the excellent talk, and well, the idea of the song is very nice too.
Let me stop sharing. We'll get to questions.
Yes, if you don't mind, we have several questions. The first one actually is from Katerina Foley, already known to you. So it's a big question. Anyway, on CARs integrating the expression and release of IL-18, is IL-18 constitutively expressed in its active form or as pro-interleukin 18, which is cleaved when the CAR T cell is activated, which may reduce cytotoxicity until CAR T cell is activated. So, and if IL-18 form is that mechanism incorporated into the CAR construct to cleave into the active form. So I'm not sure if I read it all.
Okay.
Yeah.
So this is the active form. We can detect it both in vitro and in vivo. And that correlates with what we see in terms of the preclinical studies that were conducted in vitro and what we're seeing in vivo. So that this is the active form. As far as the mechanism of the construct, I'd have to go back and ask the designers that.
Okay. The next question is from Ashlyn O'Brien, who's my PhD student actually. So she's asking, what is, in your opinion, the future of CAR and K cells?
Well, CAR and K cells, I didn't explicitly mention those, but we do have a laboratory here called the Think Lab developing CAR and K cells. And that's a potential allogeneic approach. The challenge has been, some of you know the work from MD Anderson of Katie Resvani has been the scale up. Cord blood gives you the best NK cells, and how you get enough to treat enough patients has been the challenge. But there definitely is activity, and we'll have to see what comes out of the next clinical trials. And another approach with another cell type is gamma delta T cells, another potential allogeneic approach, and there's some activity there as well.
Thank you. So the next question is from Kira Gavin. Thank you for the great talk. Did I understand correctly at the end that there are plans to charge more for the drug based on predicted effectiveness?
No, I'm sorry if I misspoke on that. So what value-based payment means is that the price is set based on a response. And if the response is shorter, or if there is no response, then there is less of a charge for the drug. So that's the principle. Now, it's difficult to implement that in the US. In Japan, I know that Novartis has applied for a mechanism like that, and that is one way to think about, okay, for an insurance system, whether it's a government payer or a private payer, why should they cover if they're paying for patients who are not responding, and it's a quite large price tag? So value-based payment is just response or no response.
Okay, thank you very much. Just going back to CAR and K cells, there was local attempt to produce CAR and K cells for myeloma particularly. And we know that from some commercial CAR T cells, like cilta cells, there is a signal of secondary myelodysplastic syndrome in patients who have been treated with that. So can you comment on that, on secondary hematological malignancies post CAR T cells or CAR and K cells, if you have come across that?
Sure. And I have a whole talk on that, but wanted to leave time for questions. So in November 2023, the FDA announced that they were investigating reports of secondary malignancies post-CAR T administration. And this was a surprise. I mean, it was a theoretical possibility that the lentiviral vector or retroviral vector through genotoxicity could induce a secondary cancer, but it had never been seen in a reliable model. So there was not a lot of information provided in the announcement of that investigation. Now, it turns out that those reports are quite rare, and they seem to be associated with pre-existing genotoxic events that patients had seen either through their prior radiation or chemotherapy or the manifestations of their disease. And there is a bidirectional risk between B-cell malignancies and T-cell malignancies. We at Penn and Stanford and other groups have published on cohort studies and case reports. And again, things break down into either it's an EBV-driven T-cell lymphoma, or there are pre-existing hits that are exacerbated by the whole treatment regimen, meaning the preconditioning chemotherapy, the significant aplasia, the inflammation associated with the administration of CAR T cells. The FDA in early 2024 did come out with a so-called black box warning. I don't know what the equivalent is in the EMA. And in some recent interviews, the director of that center has said more or less, well, maybe we would be open to reconsidering that. And this is, I think, a few things come along with this. Gene therapy has always been treated special in a not so special way, right? It's new technology. There's some underlying fear and uncertainty and caution that comes along with it. And here we have the development of secondary malignancies that many of these patients may well have developed anyway. And when you have the new therapeutic option of CAR T cells available and patients are living longer, they have the opportunity to develop secondary malignancies that they would not have had the opportunity to develop if they had passed away from their disease. So in other words, that's a survival bias. If you survive longer, then you have the opportunity to get hit by a bus. So that's not to say that it could never happen, but we haven't really seen a smoking gun. There have been reports of integrations into certain genes, but those have not been oncogenes. So we could say case is still open, but it looks almost certain that almost all of these appear to be due to other causes.
Okay, thank you very much, Bruce. We're running out of time, so thank you for very interesting, great talk. And just to announce that the next session of our cancer seminar series will be on the February 10th. We have two speakers, Josephine Haggerty from UCC on solution for implementation of survivorship care, and Jerry Lee of UCC on assessing and reducing treatment-related cardiovascular toxicity. This talk, just to remind again, has been recorded, and you can access it anytime or recommend it to whomever missed it. Thank you, Bruce, again, and goodbye, everyone.
Thank you.
Bye.